Okay, folks, if you don't mind, I'm going to get going. Today's lecture is going to be a little bit short, and so will the lecture on Thursday. My job now is to start winding the course down and to try to get you ready for the final exam. And the last thing that I need to cover with you, sort of a new technology that I'd like you to be familiar with that you will be encountering in some of your other courses in fourth year. You might be encountering it down the road in other sort of research settings, and it's important as a neurobiologist for you to be familiar with this. And I wanted to give you an idea as to why all of this was really important. We had begun our sort of discussion about neuroscience sort of through the eyes of various Nobel Prize laureates, and that's sort of where we began. And I took a little bit of a detour, and we started talking about some of the other aspects of neuroscience that are really important currently. And I talked to you a little bit about optogenetics, and it's very likely that Carl Dieseroth will be at some point considered seriously for a Nobel Prize. The field of optogenetics is something that actually is now almost a standard technique in most major labs, and it's becoming something that is increasingly popular here at the University of Toronto. A few years ago, there were maybe 1 or 2 labs that were looking at optogenetics, and now still not a lot of labs, but we have probably on the order of about 8 to 10 labs that are using optogenetics in a variety of different ways. And in the same way that optogenetics through Carl Dieseroth and Ed Boyden were revolutionizing neurobiology today, and the way that we look at behavior beyond electrophysiology from many, many years ago. There's another technique that is sort of taken cellular molecular biology and neuroscience by storm, and this is called CRISPR-Cas9, and I'm going to go through that with you today. Do you have any idea who this individual here is? Any idea at all? Any guesses? It is not my younger brother. Any ideas? So actually, his story is one of the most interesting stories. You won't hear a lot about it, but as a graduate student, he actually worked with Carl Dieseroth as well as Ed Boyden. He helped to develop optogenetics. In addition, Fang Zhang also has been at the forefront of CRISPR technology. He was one of the first individuals to actually examine talons, and then he started to look at CRISPR. Why am I saying that this is a self-serving lecture? Somewhere among all of you, some of you might be thinking about different aspects of neurobiology. You might go into clinical practice, you might stay in research, but some of you might be passionate enough about it, just like Fang Zhang was, to maybe think about doing things in a different way. And so he's actually at the intersection of 2 of the most powerful techniques in modern neuroscience today. And he had the sort of same ideas that you had when you were sitting right there. And I'm hoping that at least 1 or 2 of you might be thinking about a career in research or in clinical practice, because remember, Carl Dieseroth's a clinician. He wasn't really a scientist to begin with. And somewhere in one of the classes, I'm hoping that we have someone who has the same sort of vision for changing the way that we do neuroscience today. And in 10 years' time, maybe I'll be talking about all of your great contributions in this course. So I'm going to go through this area of genome editing or gene editing, and to give you an overview, a really quick one, of the different techniques that are available right now, and why there are some caveats to using some techniques, and why we think CRISPR-Cas9 is sort of a superior technique to be able to use. There are still lots and lots of caveats to CRISPR-Cas9. So just again, to put this in perspective for you, CRISPR-Cas9 that you've been hearing about off and on in many of your courses, the first paper in a mammalian system came out in January 2013. It is only 2 years old, and yet everyone is saying that this technique is the most powerful technique that's ever come along in the last 50 years. So we'll talk a little bit about some of these caveats, and then I'll give you an overview of what we're going to be talking about and introducing Fang Zhang's paper for the next class. So many of you have heard about genome editing and gene therapy in a number of different courses, and right now a lot of you have heard about gene editing, and you've taken courses in genetics, and you've heard about how the future everyone is going to have like a full head of hair, everyone will be blue-eyed and blonde and of a certain height. And that future, by the way, is something that the European scientists have actually come out last week with a series of papers saying that we should stop using CRISPR-Cas9 because we don't understand the technique. And at some point in the future, it might be a reality where everyone starts looking about the same because of the possibility of this genome editing and because it's so simple to actually use and to use it on the germline. So there are many different strategies for gene therapy, some of it beneficial, some of it are things that we've discussed in class, at least on a theoretical level. So we could potentially correct different types of genetic diseases if we know the genetic cause for things like Parkinson's. So we might be able to replace an alpha-synuclein or delete an extra copy of alpha-synuclein. There might be a disabling mutation, and we've heard about different types of mutations that occur throughout this course as well. And right now, what we're hoping that CRISPR-Cas9 can be used for, and it seems to be very effective so far, at least in the lab, is any type of infectious disease that is viral in origin, things like HIV and others. We can actually use that to take out the pathogenic genome as it's infecting different types of cells. So there are lots of potentials for therapeutic modifications of genomes, whether it's an infectious organism or whether it's your own unique genome to be able to edit it. And this is why the allure and the promise of genome editing is so pervasive right now. Everyone thinks that we should do it and it's a good idea, but the bioethicists are actually saying we should stop because we really don't know the implications of it. And we don't know what the cost benefits are going to be. Is it only going to be, just like in the movies, where rich individuals that can afford this type of technology are going to start using CRISPR-Cas9 because they have the resources and the finances to be able to do this. So again, I'm going to take you through the major timeline here, sort of in the middle with genome editing. Genome editing is not as old as you'd like to think. So this started really in 1979. And each of these data points that you see down here that you don't have to memorize or worry about at all, it's just to give you some context, represents one or two papers. And in 1979, it was sort of the first foray into looking at, can we modify an organism's genome? And once that door opened, there was a fairly long gap of about 6 to 7 years before the next sort of group of papers came out on genome editing. And there were lots of different techniques that were being used, and we'll talk about these zinc finger nucleases that involve things like talins in a few moments. And again, the timeline from 1979 to the year 2010 is actually not pervasive. It's not something that people were rushing out to do a lot of different papers on. And starting in the year 2010, people thought that the next big technology that would supersede all of this work that had gone on since 1979 was really going to be these talins. And I remember actually thinking about introducing talins in HMB300 because it was supposed to be the big topic. It was supposed to be the hot paper. It was supposed to replace optogenetics as the favorite technique in neurobiology. And of course, only a few years later, talins are no longer thought to be like the technique. It's now thought to be CRISPR-Cas9. So again, looking at CRISPR and the origins of CRISPR, starting from the very beginnings of understanding that different cyanobacteria get infected by viruses and that the cyanobacteria have an immune system like an immune-like system that allows them to ward off different types of viruses. That really began in 1987. It was a theoretical concept that was then related into something that would allow mammalian systems to make use of this brand new technology known as CRISPR-Cas9 that I'm going to share with you today. Since that time, though, since the publication, and it's considered a landmark publication now, just like Carl Diceroff's first publication on optogenetics. Since that paper came out in January 2013, the numbers of papers that have gone on, you can see the cluster and the dramatic increase that we get in the numbers of papers that are being published on genome editing have skyrocketed. And most labs are now doing this. And as I mentioned to some of you before this class began, it's very likely in the near future that this will be a third-year undergraduate lab. That's how easy the technique is to actually do. That's how strong this technique is, and there are already discussions about including these in third-year labs because of the strength of this particular technique. I'm going to take you through these different technologies in a moment, whether they are the original sort of zinc finger nucleases, which you can see are very similar to these talins. In fact, there really isn't much of a difference. The enzyme that is used to cut DNA here is FOC1. It's exactly the same with the zinc finger nuclease. There's different guide molecules that are involved in this process, but you can see that there's a dramatic difference, or maybe not a dramatic difference, with this CRISPR-Cas9 system. We don't have this sort of pairing of two different nucleases. We have this one nuclease that actually targets both strands of DNA for editing. And we don't need to have these guide molecules or guide modules that consist of proteins or peptides. We have guide RNA. So it's a lot easier to synthesize RNA than it is to synthesize amino acids. And we'll be talking more about this as we go along. So again, traditionally, when we started using this targeted genome editing approach, a number of different programmable nucleases, in other words, we could direct these nucleases to specific segments on DNA, such as zinc finger nucleases, abbreviated as ZFNs, or transcription activated like effector nucleases, or TALENs, and that's the acronym for what TALEN stands for, allow you to do a number of different manipulations on a number of different parts of the genome in a very site-specific manner. So this again allows you to go in and inactivate a gene. It can take out a gene that might be a mutation. It might allow you to take out an infectious genome using TALENs. You could completely delete the sequence. You could actually do element replacement or rearrangement, and you can also do chromosomal rearrangement. None of this is actually new. In fact, the technology existed way back in the 1980s to be able to do all of these, but it took a tremendous amount of time and effort to be able to do so. So again, it's incredibly useful. Right now, because of the way that it's rapidly evolving, there's a lot of thought that the way that we create stem cells now is going to be dramatically different in about 2 or 3 years' time. The technology is still going through some beta testing. It works, but it still takes a lot of time and effort to get it to work. And so having these newer systems coming out there, simpler to use, that even an undergrad could do after learning about it for 1 or 2 weeks, is something that is incredibly powerful. So these gene therapy tools have some sort of caveats associated with them as well. So there are some of these unwanted side effects. You might have insertional mutations. It might be off target. You'll hear this a lot throughout today's lecture. So these insertional mutations and the resulting non-physical expression of proteins that we don't want because of these insertional mutations might be something that even now we have to take into consideration. So these newer RNA-guided genome technologies known as CRISPR, which stands for these clustered, regularly interspaced short palindromic repeats, and the CRISPR-associated nuclease, which is the Cas9. So all of these together are a new generation of these programmable nucleases, and I'll show you why they're a little bit different in the next few slides. There are some advantages, but I'd also like you to be aware of some of the limitations that we currently have on CRISPR technology. As I said, it's very, very simple to use. Undergraduates in third-year labs will be designing these, and it might be even introduced into second-year labs. But at some point, you have to understand how this all works. I'd shown you this slide earlier on at the very top, and we haven't talked about all of the different uses that the LOXP system or the CRELOX system has. I'd introduced the ones up here to you, where you could have inversion, for example, as well as deletion. And you should certainly make sure for the final exam that you are comfortable with using either of those. There are other things that Cree-locks can be actually used for, and it has been used in the past for this. So the Cree-lock system has been used for translocation. You could have two different chromosomes nearby. You can actually cause reciprocal translocation between different elements of nearby genes by using Cree-locks as well. And you can get portions of one gene now being targeted to another region on another gene. And that, again, the Cree-lock system requires you being able to engineer and put in these LOXP sites. These are not endogenous. They're not normally occurring. You have to engineer these in. You still have to somehow get the Cree recombinase into the area that you want to have this sort of rearrangement, deletion, or inversion occurring. And so there's a lot of technical issues with that, and we've talked about some of those throughout this course as well. And again, just as a sort of a heads up for you when you're studying for the final exam, that you should definitely make sure that you are comfortable with this Cree-lock system, and especially the insertion and deletion that we've talked about throughout this course. As I mentioned to you, in 2010, this was sort of a watershed year. TALENs sort of rose up on the horizon. It sort of came out of almost nowhere. People started to suddenly be publishing papers related to TALENs, or these tau effector nucleases, and these are actually kind of an interesting technology. So they require these FOC1 enzymes, and you have to engineer it so that FOC1 gets expressed. You need 2 copies of this. You have to have it directed at a specific DNA region, and as a result, you need to have these different DNA binding motifs that are specifically guiding these FOC1 enzymes to the particular region that you want to have deleted. Usually, again, you have this DNA cleavage at that particular site through that FOC1 enzyme. It cuts. It's almost symmetrical in its cutting, and again, it requires this dimerization of FOC1 in order for it to cut at the site that you want to have removed. So you would have to know the sequences that are here. You would have to design amino acids that will be expressed and allow binding to the DNA region here. And again, that requires a lot of time as well as effort. It's worth it in the end because this technology allows for high resolution of the target sequence. You can get into the target sequence and almost with 100% certainty, if you have designed this right, it will only target that one sequence. It will not have any off targets. It won't target other sequences. And again, there's low toxicity associated with this as well, similar with CRISPR. I don't know how familiar you are with the Cas9 enzyme, but the Cas9 enzyme actually has 2 different restriction enzyme sites as well. So unlike the FOC1, where you see 2 different enzymes being targeted, it's exactly the same with the Cas9 system. It actually has 2 different enzyme sites for the 2 different strands of DNA that are being targeted. In this case, we're using a guide RNA that you have to be able to design, and this guide RNA again has this one-to-one binding with the DNA sequence, so you have to know a little bit about it. And again, it seems to be very, very specific, as long as it's designed correctly. And I'll get into that sort of at the second to last slide today. Do you have any questions about this before we go on? Have you heard about all of these different techniques in different courses? Some of these different techniques, at least Cree-locks you have, right? And again, right now, this is sort of the technique that everyone is looking at. So this CRISPR-Cas9 system is sort of interesting because previously you had to insert like a sequence, like a Cree-locks, the Cree sequence into the genome in order to be able to target this. Lots of unwanted side effects from that. So this newer technique actually allows for some advantages that I'll get to in a moment. So this is important. This is a slide that you probably want to come back and re-examine on a couple of different levels. So the core of this system really is exactly the same. We have a sequence that we want to sort of target, and we want to be able to eliminate this sequence. We want to get rid of it. Maybe there is a mutation here that is causing some sort of deleterious effects that might cause Parkinson's disease, or it might be something related to Alzheimer's disease. And we want to be able to therapeutically target this particular region and then have this Cas9 enzyme cutting here so that this gene no longer is being effective. And again, there are two different catalytic domains. There's this RUB-C at the very top, so this cuts one strand, and then there's this other strand down here, or this other catalytic site known as the HNH portion of the Cas9 enzyme that will target specific sequences. So in many ways, it's like FOC1. You need to have targeted deletion occurring at the same time. So you need two copies of FOC1. Here, it's all built into this one Cas9 enzyme. So there's one catalytic domain here built into Cas9, and there's another catalytic domain down here. This catalytic domain down here is actually targeting this guide RNA. And this guide RNA is something that you have to be able to design. It has to be something that has specificity for whatever sequence is here. I'm going to talk to you about this region that gets also really important in the discussion of how you create this guide sequence in a few moments. Do you have any questions before we go on with this? Have you heard about this before at all? Some of you have? Okay, great. So nowadays, it's a lot easier than it was back in January of 2013. In 2013, you had to clone all of these different elements together. You had to clone this CRISPR RNA, all of the yellow segment. You had to also clone and put together this transacting CRISPR RNA, the TRA-CR RNA, and all of this had to be together. Now it's all ready and packaged for you. The only thing you have to design is this guide sequence, and you have to put that in with everything that is already designed for you. So this site specificity that I mentioned to you is really important. The site specificity is because we have this complementary interaction between a guide RNA that you are designing that is being targeted by this Cas9 enzyme, as well as the presence of what's known as a PAM sequence on the opposite strand. So this is the target sequence that's going in, but this target sequence is actually derived from understanding what occurs here at this protospacer adjacent motif. It's on the opposite strand. And as a result of this protospacer that you have to have, it's endogenous to your DNA. You do not put it in. It's found in your DNA. In other words, can you think of what might be a disadvantage of this CRISPR technology already? Can I target any old gene? I just told you that this is like the greatest thing ever, right? And is it the greatest thing ever? I can only target genes that actually have this protospacer adjacent motif. If my DNA sequence does not have it, I can't really target it right now. And again, that seems to be one of the big caveats. Without this protospacer adjacent motif that's found endogenously in genes, you have to be able to search your genome, you have to be able to find it, then you have to create like a guide RNA that complements this. Without that in your sequence, you can't target this. CRISPR will not work if that PAM sequence is not there. And that seems to be one of the biggest caveats. So again, we have to be able to design this. There are lots of advantages. We don't need to design amino acids. We don't have to design all of these little guide proteins that target FOC1 to specific elements of DNA, and we don't need these bulky guide proteins. The other thing that makes this CRISPR really unique is what's known as multiplexing. So multiplexing means that you can actually target multiple regions on a genomic region. So it's not just one gene at a time. You can do this for many, many different regions as long as that PAM sequence or that PAM motif is there. And you can do all of this in parallel. It's difficult to do this TALEN technique in parallel. Usually you have to do it one at a time, which is very costly and it's very time consuming to be able to do this. This, you can just dump it on your cells, and if you have multiple regions on a genomic loci that you, or locus that you want to target, they're all sort of attacked and they're all sort of edited at the same time through this multiplexing technology. However, with all of that, this is still not the best, not always the best technique to be able to use. We have this PAM sequence that has to be there. It's endogenous in the genomic sequence that we have to be able to target. Along with that, and very important for you to realize, so again, this is a slide that you probably want to circle, make sure that you come back to, make sure that you understand. If you go back a few slides, and I didn't mention this at the time, if you're going back and looking at this in a little bit more detail, I think it's on your slide. One of the things that you'll see here is that this PAM sequence is unique and it is endogenous to your genome or to whatever genome you're attacking. And in that PAM sequence, you actually have to have a guanine residue, which is why you see this in many of the papers that you'll be reading. Without this guanine residue, before you begin, so this would be your sequence, this N would be the start of this guide sequence. So you might have a whole bunch of these guide sequences, and then at the end of your guide sequence, within your PAM, you need to have sort of this guanine residue. And that's a big limitation as well right now in terms of the technology. So this guanine at the 5' end is sort of a rate limiting step. You can't target every single genome. You have to have the PAM sequence with that 5' guanine there in order for this to work. The talins seem to be a little bit more easy to manipulate in terms of having a thymine at the 5' end. And thymine tends to be a little bit more ubiquitous, and it's a lot more common to find a thymine at the 5' end than it is to find a guanine. So that's one of the reasons why CRISPR, even though it's really powerful, really quick, and you can use RNA, there are some limitations to that. There were initial reports, and this is changing really rapidly. Fang Zhang is actually one of the leaders in this to eliminate these off-target effects. But originally, after the 2013 paper came out, and this was actually performed here at the University of Toronto in the Faculty of Pharmacy, they wanted to know, like, if you compare these two different techniques, looking at exactly the same genome, what are sort of the off-target effects? If I target gene X, do I see any other inactivation in any of the other genes? And originally, talins were much more specific than CRISPR, but the technology has caught up, and right now they're about the same in terms of the off-target effects. Again, talins can also, right now, and we can't do this with CRISPR technology, talins can also be re-engineered to target mitochondrial DNA. CRISPR cannot target right now mitochondrial DNA in patient-derived cells. So there are mitochondrial diseases that we simply can't target right now using CRISPR, but talins are able to do so. And again, it's theoretically possible that someone will be able to come up with a workaround. Maybe one of you are thinking about this, and maybe one of you will have a way to get past this sort of limitation on mitochondrial DNA right now. A lot of you might be thinking, you know, in other courses, we've already heard a lot about different forms of these long non-coding RNAs and these RNAi technologies. They were also touted to be like the next big therapeutic thing. Why can't we simply go in and use RNA interference or RNAi? Again, it's really popular. It's still used. Again, it should be something that you're familiar with using in some of your different research projects or in your undergrad labs. The one thing I'll draw your attention to, if it's never been clear for you, the effects of RNAi are temporary. They don't last. There are also non-specific elements associated with RNAi, similar off-target effects, in other words, and it's limited right now to knocking down transcribed genes. So any of the non-transcribed genes, the regions that might be regulatory elements, the intronic regions that may or may not have an effect on whether or not a gene is transcribed, has not been targeted well with RNAi. It's only the transcribed genes, the ones that get expressed, that can be targeted right now with RNAi. Again, CRISPR can last for long periods of time. It can induce both gain of function and loss of function. You can take things away, you can add new mutations back in. Right now, there are less of these off-target effects compared to RNAi pathways, which again have what's known as slippage. So the RNAi doesn't really target the right sort of sequences, and it slips a little bit, and we get sort of off-target effects there as well. You guys all take second year bio, right? So you all know the structure of RNA. So you know about these hairpin structures, and again, these guide RNAs, all of these things, you have to take this into consideration as well. So if you're creating this guide RNA, so remember, this is sort of the standard. Everyone has this now. It's pre-packaged for you. It's the sort of the guide RNA that you don't have to design at all. This is the guide RNA that you have to design. And again, it might be surprising to you, but this guide RNA has to be a certain length right now. It has to be somewhere between 17 and 24 base pairs. You want it to be as long as possible, but if it's too short, you might have potential off-target effects. It might not be targeting the right region of DNA. If it gets too long, beyond 24 base pairs, then you have to start worrying about these hairpin structures that might start forming. And so we're really restricted right now. There is a limit to what we have. In addition to that, to make this the most effective guide RNA possible, the GC content, the number of G and C base pairs that are found in this segment, as well as in the total guide RNA, have to be somewhere between 40 to 80%. It's a wide range, I know, but there is a limitation on the number of GCs as well. If you're really lucky and you've created a guide sequence that targets the PAM on the opposite strand, and you have this GC content, and it's the right length, and you are able to introduce it into a vector, like a viral vector, like AAV, or you introduce it into a lentiviral vector, it's possible for you to inject this directly into an animal and then to see what effects it has if you've designed it in the right way. Some of the really interesting technologies that are coming out are sort of these nanoparticles, and that again will probably impact you as not as students, but probably as future researchers or clinicians. So right now, nanomedicine is just starting, but nanomedicine has a lot of promise as well. You can package all of these guide RNAs as well as the Cas9 into nanoparticles and directly inject proteins now into different regions of the brain, or you can inject in the guide RNAs, etc. You can put these into a Cas9 mouse. Cas9 mice have also been generated. They have Cas9 being expressed in all of their cells, and so just by putting in this guide RNA, it's enough to get the process going as long as it's being targeted to the right cells. So how do you target it to the right cells, especially for things like neuroscience? Well, one of the things that you have to take into consideration is what is the promoter that you're using, right? So if the promoter is correct and it's being targeted to the right types of cells, you might get expression only in neurons. What type of promoter might we use just to target it toward neurons? Do you guys remember it all? And anyone remember? Is it everyone's drawing a blank on Tuesday? Think about synaptic proteins. Still drawing a blank. No ideas at all? How about, yes, Jessica? You could do beta-3 tubulin on some cells, you could, sure. How about I want to ubiquitously express it in every single neuron that I want this guide RNA or this Cas9 to be expressed in. How could I do that? Yes. New N potentially could work, but I want a synaptic protein. Any ideas? Without looking it up? Without your good friend Google, who won't be there beside you on your final exam? Yes, Victor. Synaptotagmin, close. So we had talked about the synapsin 1 promoter, and the synapsin 1 would target it toward all neurons. And again, you might do it that way, and we'll see how this was done in a paper that I'll introduce to you on Thursday. So whatever promoter you have, if you don't have a Cas9 mouse, you would express this in a cell-specific manner using Cas9. It would drive Cas9 only in those cells. Can those of you who are in molecular genetics or molecular biology, can you tell me what the function of this U6 is? It's all together. It's all in the one. It's all under one promoter. So what's spliceosome? So what's that good for? So what do you need to cut introns out of? Or what do you need to splice out of things? Like what's the molecule? Never do this to your prof. Um, any ideas? So the U6, remember RNA3 polymerase? So again, it makes sense that if this is your guide RNA that you want, that you would be using a RNA polymerase promoter. So the U6 drives the expression of a RNA expression. And again, it's really important to consider that you have cell specificity that will drive Cas9. If you don't have these Cas9 mice around, you would have this being transcribed and translated, and then you would drive the expression of these guide RNAs, which are really, really small. So you don't need to have a massive promoter. And this promoter, the U6, is normally there to drive the expression of RNA in your mammalian cells anyways. So again, consider all of this for the next paper that we'll be talking about on Thursday. And I'll go over just the broad details of it. You won't have to worry too much about all of the details. It's a fairly lengthy paper, and it's a very technical paper, but I wanted to introduce you to this technology beforehand. How many papers do you think are out there right now in neuroscience related to CRISPR-Cas9? Hundreds? Thousands? There are 5. There are only 5. And this is the only paper that came out. It was actually submitted for publication last October. It only came out in January of 2015. This is the only paper out of all of the papers, out of the 5 total that you can find on PubMed. This is the only paper that was actually done in vivo, in a mammalian brain. All of the other ones are either done in slice preparations, they're either done in cultured neurons. This is technically the most challenging paper you could possibly do. And again, I introduced Fang Zhang to you at the very outset. So he sort of sits at the nexus of all of these different technologies. He was there at the beginning to help develop optogenetics. He's there right now developing this technology related to genome editing. And what his goal is to use optogenetics to drive genome editing using mobile flashes of light that will just be traversing your brain and flashing whenever it needs to at specific regions. So he has a lot of really good ideas. And that's one of the great things about neuroscience, is that if you have a lot of great ideas and you have the ability to work through them, even though they might be really difficult, you can actually create your own sort of technology, your own field. None of this existed before, and he was able to help derive and to create this brand new sort of science related to genome editing, optogenetics, and everything else. By all accounts, he was a really, really smart individual all the way through his undergrad and through grad school. But as he relates back to some of his colleagues, you know, he often got really frustrated in his undergrad. So if you're feeling frustrated, you know, think back to Fang Zhang, who was frustrated in undergrad, had a lot of ideas that people kept shooting down, like it's impossible, we'll never be able to do it because it doesn't exist. But he persisted, and he was really insistent that he could make it work. And he has. And it's something that, again, is a moral for all of you as well, that no matter how many times someone might tell you that it's impossible, as long as you think it's possible, and as long as you're willing to put in the time and effort, like Fang Zhang, it might be something that could lead to some exciting new discoveries. And we'll talk a little bit more about his paper on Thursday. Thursday is going to be a little bit busy, so I'll be wrapping up the course. I'll be taking any questions. I'll take you through your final exam preparations. check the announcements on Blackboard. There is a sign up link on Blackboard right now for individuals to come in next Tuesday and Thursday. And the following Tuesday and Thursday, there are 2 dates for reviews for the class, the Friday before the final and then the Monday before the final exam. And I will be having some extra office hours beyond that. And it'll be a hectic Thursday. We're going to try, if the weather permits, to head over to Sid Smith and maybe try taking a group photo as well. So we're going to try ending a little bit early, somewhere after 10:15. So be prepared to, one, look good. You always look good. And two, to have somewhat of a disjointed class that way. Okay?